Hello, dear students. I hope you all are fine. I welcome you all to Sri Jayendra Rangjun classes. I am friend and mentor for coming classes with respect to botany part of biology. And in this lecture, we are starting with the one of the important chapter of class 12th, that is sexual reproduction in flowering plant. So, dear students, by hearing a word flowering plant, you came to know about a angiosperm, and these are the only plant which bear flower. And you know the flower has too much importance in our life as well. We offer it to the God, to our near ones, and that is one of the essential part, and it is also have some of the economical role. So in this chapter, we are not going to deal that all aspect. We are mainly going to deal with the sexual reproduction part. Okay. So in reproduction part, first we are going to introduce some of the things in this, particularly with respect to this chapter. So, dear students, you know that all flowering plants show sexual reproduction, or particular angiosperm shows sexual reproduction, and the capability of sexual reproduction is gained after a certain period of life in case of plant. Now, if we talk about angiosperm, the structure of inflorescence means it is a group of flowers in which several flowers are arranged on a floral axis. Then it is known as arrangement of flower on a floral axis, that is inflorescence. The structure of a flower and other floral plants that give an amazing range of adaptations that ensures formation of end products of sexual reproduction. And what are the end products of the sexual reproduction? Fruits and seeds. So here, sexual reproduction is mainly help us to understand how fruits and seeds are produced. Understood? So, dear students, we are going to deal with in a very well manner. You'll see several different types of plants and different type of fruits can be present. And this give rise to the seed. You can see this is the new structure of seed, and the seed has a seed coat, embryo, cotyledon, and seed. That seed is able to give rise to the new plant. Apart from dates, you know that what do you mean by embryology? That is also we are going to study in this part. And embryology is a study of embryo, or in that also we will talk about embryogeny as well, as that is embryo formation. And here, this is a branch of botany that deals with the nature and development of the structure and the process associated with sexual reproduction in angiosperm. Okay, so it is dealing with the particular processes associated with sexual reproduction in angiosperm. Now, in this, when we study the sexual reproduction, the major thing or major organ of the angiosperm, that is a flower, which is playing major role in the particular sexual reproduction. So, flower is a fascinating organ of angiosperms. Flower is a fascinating organ of angiosperm. And you know that flower are object of aesthetic, social, religious, and cultural value. You know that we give it to the god or goddess. We use it in a social functions, and we had has so many cultural values. We used it also in a decoration purposes. So these are the different roles of this flower. Now, when we talk about a reproduction, to a biologist, flower is a modified condensed shoot. See, in the shoot tip of shoot, you will find initially the apical meristem, and that get modified into the floral meristem, and the floral meristem will form what? Flower, and that flower performs sexual reproduction in angiosperm. So, if we talk about this is the flower, and in typical angiospermic flower, mainly when we see the flower, it has a four walls of floral appendages on a receptacle. On the basis, on the thalamus, you will find the presence of four walls. That is a calyx, corolla, androecium, and gynoecium. See, this is the structure of flower. You can see here the green colored receptacle is there over which the sepals, green colored sepals are arranged. Then apart from this, you can see the another colored petals are there. Then filament and anther are there, which forming the stamen, and the style, stigma, and ovary that forming a carpel. So the greenish sepals are there, and the group of sepal is known as calyx. Group of petal is known as corolla. Group of stamen is known as androecium, and group of carpel is known as gynoecium. So that four walls are forming a particular structure of this flower, and this is a beautiful structure of flower is obtained. So dear students, in this we have studied just now that flower has a particularly two walls. One is non-essential organs and the another one essential organs. Now non-essential organs are the accessory or helping walls or perianth members, and essential organs are the reproductive wall. So in a non-essential organs are the sterile parts, and which includes the calyx and corolla. And calyx, you know, that is a group of sepal which we have just studied in the diagram, and the corolla is a group of petals. So these are not playing a role in a reproduction, so they are sterile. But when we talk about essential organs, in essential organs you will find that there is a presence of androecium and gynoecium. Now, dear students, these two have main importance in sexual reproduction because androecium have a group. It is a group of stamen, and stamen will produce a male pollen grain, and pollen grain has a male gamete. Whereas gynoecium or carpel have an ovary, and in ovary, particularly, you will find a presence of what? A ovule, where a egg formation occurs. Apart from this structural part of this flower, here we have to talk about the sexual reproduction by classifying it in the different events like pre-fertilization event, fertilization event, and post-fertilization event. Okay, so here first we are going to talk about pre-fertilization event, and then we will talk about the fertilization event. So here we are going to discuss pre-fertilization structure and events. Now, pre-fertilization structure and events, when we talk, it include mainly stamen, and in stamen you will find a microsporangium, and in microsporangium the pollen grains are produced. So stamen is also considered as a microsporophyll, and it is a male sex organ, and male sex organ has, or a stamen has, a two parts, and the two parts are the what, particularly we can say anther and the filament. Now this is the anther and this is the filament. Now apart from this anther, if you look, it is a broad knob like fertile part. It is a fertile part. And when we talk about angiospermic anther, it has a two lobe. In previous diagram also you can see there is a presence of two lobe in one anther. So when two lobe are there, it is named as what, a dithecus we can say. But see, in each lobe also you will find a particularly two pollen sac. In one lobe there is a two pollen sac. So in total two lobe there is a four pollen sac. Okay, so say this way, particularly four pollen sac are found. So this type of anther will be considered as a tetrasporangiate and bilobed. Now here one more thing you can see in this diagram, that there is an anther and there is a filament. The attachment structure or a region of filament when or region of anther where filament attaches, that is known as connective. We remember connective joins the what, anther and the filament. Then this is the structural part which is showing here. You can see the another cross section, and in cross section you can see the microsporangium is there. The last cross section you can see microsporangium, that is a cross section of anther, and there is a microsporangium, that is a pollen sac. Four pollen sac are there, and in pollen sac microspores are produced. Now the dithecous anther consisting of four microsporangia, two in each lobe. Actually, the term dithecous is used for, it means the having a two lobe, two pollen sac in one lobe, that is found in case of the datura. So this datura have a particularly four microsporangium, two in each lobe. Then this is the structure of particularly flower. Okay, datura. Then see, this is the actual diagram. You can see this is a cross section of anther that is having a what, microspore pollen grain which are produced in the pollen sac. Okay, and connective is there. See, this is the region where the filament attaches. Now, students, after seeing this floral diagram, several floral diagrams, next we are going to talk about the one of the monothecous anther. Monothecous anther means consist of two microsporangia in one lobe. Okay, like the hibiscus. Okay, in particularly one lobe, the two microsporangium are there. So this is the monothecous one, and it is found in case of the hibiscus one. Now you can see this is the particular diagram of this one. Apart from this, next we talk about the filament, and filament, it is attached to the what, a particular anther, and it is a sterile long slender stalk. Now this is the one of the important question which you should focus here. The end through which the particular filament is attached to the anther is a distal end, and the end of the filament which is attached to thalamus or petal or tepal, that is known as proximal end. Means the near one is a proximal end and far away is the distal end. So you can see here in diagram, you can see this is the filament, and you can see the such type of filament are found in case of this one. So one question we are going to discuss, that is the male sex organ in flower is called stamen, carpel, petal, and sepal, and you know that it is a stamen. Apart from this, next another question is that a broad knob-like fertile part of stamen is called. So you know that filament cannot be there, carpel can be there, petal cannot be there, and it can be the anther. So anther is a broad knob-like fertile part of stamen, but filament is not a fertile part. Then third we can see monothecous anther is seen in. So just we have discussed hibiscus is the answer for this. Then next we talk about the structure of microsporangium and the layers of their wall. So in this, when we structure or transfer section of anther we take, we particularly see where the homogeneous mass of a meristematic cell, that is called a primary sporogenous tissue in case of younger anther. And this primary sporogenous cells surrounded by anther wall. Okay, and primary sporogenous tissue, if you see, that are going to form a microspore mother cell inside the microsporangium. So see, so these are the homogeneous mass of what, initial cells that are going to form what, particularly microsporangium. Okay, these are particularly forming a what, microspore mother cell inside a microsporangium. Now, when sporogenous cells give rise to the microspore mother cell, that will further going to divide and form a microspore. So this is the TS of microsporangium and mature dehiscence anther. So pollen grains are getting released. Now first we are going to talk about anther wall layer, then we will discuss what actually is going to happen in the pollen sac. Now, when we talk about anther wall layer consists of four layers. First is epidermis, and epidermis is the outer one celled thick layer, outermost single one celled thick layer, single celled layer is found. And epidermis is the outermost covering, so it is a thin wall present between the two pollen sac. Belonging to epidermis is called a stomium. See, the thin walled cells present between two pollen sac belonging to epidermis is called a stomium, and when stomium cells broken up, the pollen sac get dehisced and pollen grain are released. Okay, so you can see this is the epidermis outermost covering, and the stomium is the particular region between the two pollen sac which belong to epidermis. Then we next talk about the endothecium. Dear students, you know that endothecium is the second layer. Inner to the epidermis, you will find the second layer that is known as endothecium. Now, the first statement that is important one, that the cells of this layer have a cellulosic fibrous thickening. See, they have a cellulosic fibrous thickening, and that help in dehiscence of anther. That help in dehiscence of anther due to the hygroscopic nature. So they have a fibrous cellulosic ring on tangential walls. Okay, and which is also helping anther in the particularly dehiscence. See here you can see the outermost is epidermis, then endothecium is there. Then inner to endothecium we will find a middle layer, and inner to middle layer we will find a tapetum. These are the four layers. Next, third layer which we are going to discuss, that is the middle layer. Dear students, in middle layer you will find that first thing, middle layer are 1 to 3 layered, and the cells of this layer are ephemeral. Ephemeral means short lived, means they are going to degenerate at the maturity. Now when you see this cross section, you can see that middle layer are present in multiple layers. And the last we are going to talk about the tapetum. So tapetum, when we talk, tapetum is the innermost wall layer of the particularly anther wall. And when we talk about the innermost layer of this anther wall, that is a tapetum, you come to know that this is the innermost layer of anther wall which surrounds the sporogenous tissue. Next, the thing which we know, that is a tapetum nourishes the developing pollen grain. Tapetum nourishes the developing pollen grain. Okay, and tapetum, when nourishes the developing pollen grain, the cells of this tapetum possess a dense. So, because why? Pollen grains are developing within the pollen sac, and the pollen sac are covered by this tapetum. So, tapetum will provide development to the nourishing, particularly pollen grains. When we see the characteristic of the cells of tapetum, we find that the cells have a dense cytoplasm. They have more than one nucleus. They are multinucleated, and they are covering the particularly pollen sac. When we talk about the next one, the sporogenous tissue which are present within the pollen sac. So here you can understand that each cell of sporogenous tissue is capable of giving rise to the microspore tetra. Means what happens? The sporogenous tissue which give rise to the microspore mother cell. The microspore mother cell will undergo meiosis and form microspore tetra. Therefore, each cell which can act as a microspore mother cell can be called also called as a potential pollen mother. Now, next we are going to talk about particularly formation of pollen grain or microspore, and that process is known as microsporogenesis. So what is microsporogenesis? It is a process of formation of microspore from the pollen mother cell or microspore mother cell through the meiosis. So through meiosis, when microspore mother cell undergo meiosis and form microspore, it is known as microsporogenesis. Now, apart from this, the microspore which are formed are arranged in a cluster of four cells and called a microspore tetrad. You can see here one of the important and best diagram is given here. Pollen mother cell is given, which is a diploiding stage. When it undergo meiosis, it form a two haploid cells. After meiosis, two, four haploid cells are found that are known as pollen cells. But these pollen cells are found in groups, so this is known as tetrad of four haploid pollen sac. Then secretion of wall occurs and wall breakdown occurs, and particularly that pollen grains are secreted. So pollen grains are generally spherical, measuring about 25 to 50 micrometer in particular diameter. See, we are going to discuss the structure of pollen grain. So pollen grain are generally spherical shape, and they have a diameter of 25 to 50 micrometers. See, this is a so easy points, but they can ask because it is given in NCERT. Apart from this, the cell wall of pollen grain is called sporoderm, and the cell wall is made up of two layers. See, here you can see diagram. The two layers which cover the cell wall of pollen grain is called sporogram, which consists of two layers, that is known as exine and intine. Outer harder layer is called exine and inner thin layer is called intine. So here this type of pollen grains will be visible in nature, and the outermost covering is exine you will find, and the inner covering you will find that is a intine. Now, exine, if we talk, the exine, the outermost covering is made up of sporopollenin. And what is sporopollenin? Sporopollenin is nothing but a fatty substance, and it is known as the most resistant organic material on this earth. Now, this is a fatty substance, and this sporopollenin, if we talk, that cannot be degraded because it cannot be decomposed, it cannot be broken up by electric shock, even that much hard or tough it is. So here you can see this TS structure of pollen grain. In that, the exine you can locate, and apart from exine, you can also locate a germ pore, which are some of the depression in the exine. See, germ pore are what? Germ pore are the particular areas of exine where sporopollen deposition is not there. Okay, and apart from them, exine is not affected by high temperature. Okay, and strong acid or alkali, and no enzyme can degrade the because it is made up of sporopollen. Apart from exine, you can see inner the intine is there, and that intine is made up of pectocellulose. Okay, that is made up of pectocellulose. And inner you can see two type of nuclei. One cell large is there and one small is there. Small cell is known as generative cell and large cell is known as what? A vegetative cell. Okay, students. So I hope you understood the exine. Because of the presence of sporopollen, pollen grains of the past plants are well preserved as fossils, so they can be preserved as fossils also. Pollen grain exine has a prominent aperture, germ pores where sporopollen is absent, means we can say exine is a discontinuous. Okay, and another one is the intine, and that is made up of cellulose and pectin, so we call it as pectocellulosic. And if we talk about development of male gametophyte, the pollen microspore is the first cell of male gametophyte. The germination of microspores starts in situ in the mother place. Microspore may be best defined as a partially developed male gametophyte. They can be developed as partially male or developed male gametophyte. What happens in case of microspore? Microspore divide mitotically to give rise to one large vegetative cell and small generative cells. So, generative cell, which is a smaller one, which is spindle shape, it is a dense cytoplasm and a nucleus, and this way the male gametophyte development occurs. Okay, and see what happens. The pollen grain have now one vegetative cell, one generative cell, and it is known as two celled stage. But when generative cell undergo mitosis, they will form a two male gamete. So in one pollen grain you will find two male gamete and the one vegetative cell, and that will be considered as three celled stage. So pollen grain from anther mainly released in two celled stage, having a generative cell and vegetative cell in 60% of angiosperm, whereas remaining the pollen grain are released at the three celled stage with the male gamete. And the further development of male gametophyte takes place on the stigma. Now how this happens, that we are going to study, dear students. But till that, in today's lecture we are stopping here. In further lecture we will be continuing with the what particularly the female gametophyte development and formation of the egg cell as well, we will be going to study. So I hope you all understood till now. Thank you, thanks all, and we will continue it in the next class. Okay, dear students.